Good morning, everyone. So this is work that I did far back, really back in my PhD. I know a lot of things have changed in terms of transmitter founder and chronic infectious envelope. So we know that heterosexual transmission is the major means of HIV transmission, and if we understand the virus that is transmitted, today we learn that it's more than one virus. That time we thought of one virus that's transmitted. We can block that one and actually prevent infection. And so my group and other people had found that dendritic cells had played a role through DC sign binding and trans-infection. And so we were characterizing the envelope of HIV in terms of the N-glycans, the structure, and how it binds to DC sign. And we had found that it both, remember, the glycans can either shield neutralization antibody, or some of them even act as epitopes. And then we found that it induced the dendritic cells. It could bind to DC sign through dendritic cells through DC sign and induce production of IL-10, which causes deregulation of the immune response in the genital mucosa. So we look at the position of the N-glycan, the type of N-glycan, and the heterogeneity of the N-glycan in terms of how they interact with DC sign. So because we found that these are factors that could play a role in binding to DC sign, we decided to characterize their binding to specific lectins. You know, DC sign is a lectin receptor, so we said there are specific lectins that bind to specific glycans. So we decided to characterize their binding to lectins. So because we had found that the transmitter founder induced more IL-10 than the chronic infection, both in the pseudotype envelope and in the soluble envelope. And then we tried to see on the dendritic cell, you have both toll-like receptors and the C-type lectin receptors. So if we co-stimulate with other toll-like receptor agonists like the HKLM, the LPS, and the poly-IC, which is a toll-like receptor 3 agonist, we found that it could reduce the deregulation of IL-10 production through dendritic cells. So we decided to say, let's just look at what are the glycan types. So if you bind them to specific lectins like the daffodil lectin, the PHA, which has, daffodil, for example, binds more high mannose. So we could see that instead of, although the transmitter founder induces more IL-10, and we had set through DC sign, and DC sign is set to bind more high mannose, the transmitter founder in this case is not having way more high mannose than the chronic infection clones, because you could see that the chronic one, C13, binds more daffodil lectin. But when you characterize the binding to phytohemagglutinin, which is a complex type glycan binding lectin, you realize that the transmitter founder binds binds higher. This is using surface plasmon resonance binding. And then you could see that the transmitter founder has more complex glycans than the chronic infection. So then when we just decided to say, let's use PG9, which is a glycan binding monoclonal antibody, and then just look at other CD4 binding antibodies, you could see that they really do not even bind. So here to conclude, we could say that not only the types of glycan or the heterogeneity, but I think also the position where the glycan is placed on the envelope influences its binding to DC sign. So it's not just a high mannose or a complex thing, but where the PNG is placed on the envelope glycoprotein of HIV, subtype C, which we used. Thank you.